Thank you everybody for coming today. I'll be talking about our method on metagenome strain haplotyping today. This is co-led with my co-author John Sebastian Gounod, and this is a joint work between the University of Toronto and the Genome Institute of Singapore. So the theme of this talk will be about strain level heterogeneity, which is really important because let's say I have a microbiome here with just 2 E. coli strains. One could be pathogenic, one could be commensal. But when I do metagenomic sequencing, what happens is that I just kind of sequence everything and the reads are strain mixed. So if I do assembly, this is alluded to in the keynote, what happens? Do I only get the orange genome? Do I get the blue genome, or do I get both genomes? And the answer is that it depends on the technology and the algorithms. Broadly speaking, with short reads or the low fidelity long reads like nanopore simplex reads, you'll tend to get one strain using these algorithms I've listed. But with these more advanced hi-fi ASM methods, meta-MDBG, with these PacBio hi-fi reads, you can get both strains a lot of the times. So this presentation will be about asking, how do we get more information out of these cheaper and noisier sequencing technologies? And how we're going to do this is something called haplotyping or phasing. The idea and the computational goal of this presentation is really to go from reads to something I call strain level haplosets. Haplosets are clusters of reads where all the reads belong to exactly one strain. And the reason why I want to do this is because if I can cluster the reads into strain level haplosets, then I can just run an assembler downstream, or I can collect the sequence of alleles I care about, which are the haplotypes. So I want to focus on this step. How do we deconvolve the sequencing data and separate it and cluster it into strain level data sets? And we do this through using heterozygous alleles, in this case SNPs, and read linkage information. So if I have some reads, I can map the reads onto a reference genome. I can call SNPs, call variants. And the idea is if I have heterozygous alleles and reads that span more than one heterozygous allele, I can link these reads together so that they all match the same alleles. And these reads that match the same alleles will come from the same strain or the same haplotype. So I use these SNPs to cluster my reads. And that's what the general idea of this talk will be about. So I'll start off with the contributions in our work. The first part of this work is a read clustering, which I will call synonymous with phasing algorithm. It takes alignments and some SNPs and is able to output those haplosets we care about. It's able to deconvolve the sequencing data. Floria is written in Rust. There's documentation, as you can see below. It's easy to install. It's pretty fast, as we'll show. But we've also extended Floria to be integrated in a snake-make pipeline that goes from FASTQs all the way to whatever you want. Because the standalone Floria, you need VCFs or BAM files like alignments and variants. And I'll quickly go over the pipeline. The pipeline starts like this. So first, you have short or long reads. And then the first step is to generate a set of reference genomes, because I need to map, I need to call variants. So I need a set of reference genomes. And you can do that through either Kraken profiling, which is integrated in this pipeline, or you can use a metagenome assembly, where the contigs from your assembly are your reference genomes that you map to. Next, in the pipeline, we map reads and call variants, and then we can feed those variants and reads into the Floria phasing algorithm that outputs those strain level haplosets I care about, and then you can run an assembler downstream on those haplosets to get a strain level assembly. This is all integrated in this pipeline, but what I want to focus on today is this red box. It's a new Floria phasing algorithm. This is the main algorithmic innovation today. And it follows through 3 big steps. I've expanded out the Floria box here into these algorithmic steps. The first step is something called local clustering. The idea is I have my contig or a genome, and I have some map reads and I have some SNPs. The first thing I do is I break that contig into smaller chunks, these blocks, and then I cluster reads in these chunks individually, one chunk at a time. And in this case, you see there are 3 strains. There's a red, green, and blue strain. And sometimes my clustering will have 2 clusters locally. Sometimes it'll have 3 clusters locally. But how do I do clustering? That's the algorithmic question here. And we do clustering by optimizing something called the minimum error correction score. The MEC score is a measure of clustering goodness. It asks you how many alleles look like errors within the clusters. How many errors do I have to flip in order to get a good phasing? People have studied this in the context of human haplotype phasing. And we know it's NP hard to compute. So what we do in Floria is we use a beam search heuristic. You may have heard of beam search and natural language processing and decoding and things like that. It's just a greedy method of searching your space of solutions, and we integrate it for clustering in this case. But there's a thorny issue, which is we don't know the number of strains a priori, right? Like, how do we know how many things to cluster into? And the idea is to run our clustering algorithm iteratively. So you try 2 clusters, you try 3 clusters, you try 4 clusters. And once this measure of goodness called the MEC score, once it plateaus, then you know you'll have the right amount of clusters, because it'll keep increasing in goodness until you've hit the number of strains. So that's the clustering step. The main idea here is to look at the config locally and cluster things locally. And the next step is going from local to global using a graphical representation and network flows. What I've shown here is the previous local clusters that I've gotten, but I've made it into a graphical format. Each of the nodes are one of those read sets, local read sets. And I formed a directed acyclic graph, a DAG, where the edges are weighted by the number of shared reads between those clusters. Because if you remember, those blocks are overlapping. So you can have shared reads. So once I have this representation, it's kind of nice. It's a graph, but you can have errors in this graph construction. Over here, this I call an error because the coverage is 8. It looks a little too low. That green strain, judging by the side edges, should have about coverage 30. This graph does not take into account the coverage consistency that you'll have for a strain genome. So what we do is we change the edge weights to enforce network flow conditions. A network flow requires that the input edge weights are equal to the output edge weights. And this enforces coverage consistency because the coverage along a genome should be about consistent. And the way we do this is by linear programming. So what we do is we find a new flow that is similar in L1 distance, the original weights W. And we solve this using linear programming because you can encode all the constraints as linear constraints, and we show how to do that in the paper mathematically. Great. So this step takes those local clusters, embeds it into a graphical representation, and uses coverage to regularize the graph to come up with a better graph. So that's the big idea here. And the last set is to obtain haplosets by going through the graph. How do we do this? So the idea here is I have this graph. I want to find the largest minimum flow path from left to right. I want the path from left to right with the highest minimum flow. And in this case, it's this guy over here because the minimum flow is 40. So I take this path, which I can find using dynamic programming because it's a DAG. You can do dynamic programming on DAGs very easily. And you extract this path. You do this again, you extract this path. And now I'm just left with disconnected components, and I'll just take everything. And then all of these paths, the 4 paths in total, including the disconnected components, you can aggregate into a haploset by just taking all the reads within the path and taking the union of all the reads within the path, remembering that these vertices are clusters of reads. So that's the Floria algorithm. It goes from local clustering to some sort of global regularization using linear programming and network flows, and then we use some sort of dynamic programming to extract paths carefully that are good strain-level haplosets. Now I'll talk a little bit about synthetic benchmarking. So we simulated a metagenome here with 40 common gut species. We took 1 to 5 strain genomes per species, like real strain genomes, and we simulated really, really noisy nanopore reads because we wanted to see how low can we go as far as sequence identity goes. And we simulated random coverages between 5x to 25x for each of these strain genomes. So they're not uniform. And then we compared against, at the time we were writing this paper, the published state-of-the-art methods, which is Metafly for just metagenome assembly. It's not very careful at disentangling strains. But we also compared against Strainberry, which is published and is meant for disentangling strains. And what we found was this. So on the left here, this is the path in the Floria PL pipeline that we take. We use Kraken to identify genomes. We use Minimap and Longshot to align and call. And then we use Fly for downstream assembly. And we compare strain level assemblies between all 3 algorithms. In A, what you'll see is the genome aligned percentage. This is the percentage of strain genomes that we obtain. And higher is better here. So Floria looks to be doing quite well. In B, you have the N50, so larger is better, is more contiguous. And in C, it's the number of structural errors as a result of the assemblies. And here you want lower is better. D is an interesting figure. So this is the time it took for these pipelines to run. And we stratified it by the individual steps in the pipeline. Floria, the pipeline, takes the least amount of time. But I think what's interesting is that if you zoom in, you'll see this dark green, really small stripe that you can barely see. That's the time it takes for the Floria algorithm to run. Everything else is assembly time, it's mapping time, it's variant calling time. So the actual phasing algorithm itself, if you didn't want to do assembly, it's extremely fast. So now I want to move on to real results because I think they're a little bit more interesting. We have more synthetic results. We have some synthetic results on short reads, all sorts of reads. But I want to talk about some real results. This is one thing we ran. So this was 109 gut nanopore samples taken from this Singaporean cohort led by my co-author, John Sebastian. And Floria took less than 15 minutes to phase per sample. It's very fast, and variant calling was actually the bottleneck here. In figure C, we show 50 different species listed by the most prevalent species and the number of strains we found in each sample. So that was interesting, but we investigated further in part D. So what part D shows is individual phasings for specific species in samples. Each bar here is one of those haplosets, is a strain-specific read set. It's spanned by the distance along the genome. The y-axis is the coverage, and the color you don't have to worry about. But what you'll see is that the haplosets cluster by coverage. This is what we want because we know that a strain should have a consistent coverage along the genome. So this gives some credibility to our results. There's the E. coli example you'll see there. It looks a little bit more crazy. It looks like maybe there's 3 strains going on. So we investigated it further. And what you can see is that Floria allows for visualization really easily because I'm just outputting sets of reads. I can map them. I can look at them in IGV. And what you'll see is that the alleles for these 3 haplosets are consistently different. They're consistent within the haploset, but they're different across haplosets, which means there really does seem to be 3 different strains here. And we can visualize it. Another cool example is longitudinal strain tracking with Floria. So what we did was we took 24 longitudinal short read gut samples over about 2 years. This was not our data, but this is public data published in this 2003 genome biology paper by Watson et al. And what we did was we ran Floria, obtained haplosets, and we wondered if there were any strain level tracking patterns that we could find throughout almost 2 years. And we found some pretty cool stuff. There were 2 species with very, very interesting strain diversity patterns. What I'm showing in this plot here is haploset matches across time. So each line is a match between haplosets for 2 different time points. Okay, and the y-axis is the coverage. That's we took the log to stabilize variance. But what you'll see in the top figure, for example, is these 2 bands that track a long time. And each band is a set, it's a cluster of haplosets at a specific coverage. So there seems to be 2 strains that track across almost 2 years, one always high coverage and one always low coverage. On the bottom, you'll see Anaerostypes hadris, which has a much more dynamic sort of a strain tracking pattern over 2 years. And we wanted to investigate this further. The color, by the way, indicates the number of alternate alleles compared to the reference for the haplosets. So it's just a genomic signature, which you can kind of like track along. There's a sample mislabel at day 614 we found. We found this really weird collapsing of strains, and suddenly it gets okay again. And we think it's a sample mislabel. But we want to investigate this behavior further, especially for this low to high strain emergence for this bottom species. So we visualize this. On day 391 for A. hadris, there's 2 haplosets. There's a green one and a red one. The red one is super low coverage, and I had to zoom into it. On the bottom is actually a zoomed in version. On day 446, I'm showing 2 haplosets. Both of those haplosets on day 446 are high coverage. But if you look at the alleles, you can see that the alleles are exactly the same throughout both 50 days apart. So it really does seem like this strain emerged from low to high abundance, or it was replaced by a very similar strain. But what we've done is we've visualized what we see in the data. And it turns out, Floria can do this sort of thing. Okay. It turns out Floria works on ancient viral metagenomes. This is very interesting. I didn't do this work, but after Floria was published, Maxime Bory, who's a postdoc at Max Planck, he showed me that you can run Floria on a mixed infection of metagenome sequencing of 2,000-year-old samples of hepatitis B. And apparently, you can tease out the 2 different strains here, which I thought was very cool. So in conclusion, what have we done here? So we've introduced this algorithm, Floria. It's a strain-level read clustering algorithm. That's essentially at the heart of it, a strain-level read clustering algorithm using SNPs. And we've also integrated into a pipeline. It works on short or long reads. That longitudinal example with strain tracking, only short reads. Takes less than 20 minutes per metagenome, and variant calling becomes the bottleneck in this case. And you don't have to assemble things. You can track things without assembly. So what I think is interesting about Floria is that it's incredibly versatile and fast. We do benchmark on strain-level metagenomic assembly, and this is a very important problem. Like strain-level metagenomic assembly is the gold standard. It's what we want. But if you're talking about short reads or really noisy reads, it's just kind of impossible. What I hope to show you is that you can do this sort of thing even if your data quality isn't very good and still get very interesting hypotheses to generate. And I think this tool, it's versatile, it's fast, and allow you to do data sleuthing and really investigate your hypotheses much more. My final slide is acknowledgements. I'd like to thank all my co-authors, John Sebastian in particular, who's the co-lead here, and the PIs, Niranjan and William. Our paper is out now in Bioinformatics Proceedings. We'd like to thank our funding sources. And thank you very much.
Thank you. Some questions here.
Thank you for the interesting talk. I have a couple of questions about the data. So seems based on your experiments, can you say that it is very common to identify multiple strains for many bacteria species?
Yeah, I think so. I mean, I think that when you sequence it obviously depends on the biological conditions, obviously diversity and all the factors that impact diversity. I don't know. But definitely you can see a lot of strain behavior present. Absolutely.
Okay, because I think that also related to how the strains evolve, right?
Mm-hmm.
For example, RNA viruses, they naturally mutate very frequently, so you tend to see multiple viral like quasi-species. But for bacteria, I don't know whether it's common. You always see multiple strains. Yeah, but seems that's your observation. Okay, thank you.
One last question.
Well, it's kind of the same question, but I wonder about the frequency of the strains. Did you observe in many times that there is one that it is most dominant, or it should be kind of the same frequency because they are the same species and essentially are very similar?
I think from what we've observed, I would say it's more common for one to be dominant and not the same frequency, just because, I don't know, if you were just to randomly pick frequencies, they wouldn't happen to be perfectly in equilibrium, right? Yeah, I mean, I will say that this study, we didn't go too much deeper into the biology and looking at the statistics of strains and their abundances. But from what we've done in like working on the algorithm and testing, it seems like non-uniform coverage is more frequent.